Hello, it's Dr. Debbie Morris again in a t-shirt, again to talk about gastroenterology. So I did some reorganizing of this material from previous years that you might notice if you look in the notes from previous years to try to make it a little bit easier to follow. But again, I've had help from Dr. Perry, and if you have any suggestions about organization and about problems with the presentations, remember that you can always let me know. I'm not going to take it personally. I'm actually going to appreciate it. That's one of the things that I think we say in orientation, but I'd like to say again as we're getting closer to the middle end of the semester, is that your feedback really does inform how we teach here. And I think that's one of the unique things about Methodists that makes us as great as we are. So when the time comes for you to do course evaluations, I want you to be as honest as you can be while remaining very professional. So it never hurts to make suggestions and it never hurts to tell us what concerns you have. So we have left the stomach through the pyloric valve and have entered the small intestine. And today we're going to talk about a number of important issues that have to do with the small intestine. The first is about movement in the small intestine. And you remember the layers of the intestinal wall, and the classic layers with 2 layers of muscle are present in the small intestine since we've left the stomach. So there is longitudinal muscle and circular muscle. And that muscle allows the small intestine to mix the chyme and to propel the chyme forward until eventually it reaches the ileocecal valve. So mixing contractions, we talked about this a little bit yesterday. When there are portions of the intestine that become distended, there are mechanoreceptors that detect the stretch and cause contraction of circular muscle to have concentric contractions at intervals along a segment of the small intestine, which segments the chyme in the small intestine. As those contractions relax, there are circular contractions at different intervals along the small intestine. And so this chops the chyme and promotes the mixing of the chyme that has left the stomach with the secretions that are being added to it in the small intestine. There is something called electrical slow waves that determine the frequency of these contractions. The rate of slow waves, the pacemaker as such, is influenced by the enteric nervous system and signals from other parts of the intestine and locally, as well as by the autonomic nervous system. So here again we have pictures of peristalsis in which a circular contraction forms because of distension at a bolus of food, resulting in constriction, which moves along the wall of the intestine, propelling that bolus forward. And here we have mixing contractions where the mechanoreceptors detecting stretch cause interval contractions of smooth muscle, which then change from place to place, which is mixing the food and slowly propelling the food, although there is a combination of these two kinds of mixing. Peristalsis is extremely important for moving the chyme forward to new areas of small intestine wall. Peristaltic movements aren't very long. So a peristaltic wave that propels chyme forward can only travel a couple of inches before it dies out. And so chyme moves forward at about a centimeter a minute. So this is a very slow kind of motion. And the reason for that is that the chyme has to stay in the small intestine for 3 to 5 hours in order to pass out of the small intestine into the colon, so that everything that needs to be digested is digested and everything that needs to be absorbed is absorbed. The control of peristalsis, as many of the other functions of the GI tract, has to do with signals from the enteric nervous system, from the autonomic nervous system, and from hormones and from paracrines. So after a meal, peristaltic activity increases, and hormones that are involved in that include gastrin and cholecystokinin, motilin, serotonin, which, if you remember, I said was one of the neurotransmitters that is used in the enteric nervous system, and then insulin. Hormones that decrease peristaltic activity include secretin and glucagon. So insulin and glucagon, if you remember, are both pancreatic hormones that are passing out of the pancreas through blood. There is also movement of food forward from segmentation and mixing. Occasionally, there'll be something called a peristaltic rush, which is a powerful and rapid peristalsis, and we often hear that or see that on the surface of the stomach as unusual motion. The next thing I want to talk about concerning the small intestine is secretion from the small intestine. Now, earlier when I was talking about secretions, I mentioned that the primary function of the small intestine is to absorb nutrients and their digestive products. However, during the course of a day, a large amount of fluid is secreted into the chyme of the small intestine. Between the intestinal villi, and here we have villi, there are pits that are called the crypts of Lieberkühn. And on the surface of the epithelium of those pits, there are cells that secrete mucus that are called goblet cells, and these goblet cells secreting mucus help to protect and lubricate the surfaces of the intestine. And there are cells that are called enterocytes that secrete water and electrolytes from the crypts. Enterocytes also reabsorb water and electrolytes in the villi. So here we have a diagram where we see villi, we see everything covered with an epithelium, we see these pits that lead into crypts. There are goblet cells in the crypts, there are enterocytes in the crypts and at the base of the villi. There are also cells that support this, and these are very actively reproducing cells. So lots of mitosis going on because the lining of the intestine is being continuously abraded and continuously replaced. From the crypts, we have active secretion of chloride ions, and we have active secretion of bicarbonate ions. These anions coming out of cells and into the lumen of the intestine drag cations like sodium through the membrane and into the secreted fluid. And because of all of the movement of these electrolytes, we also have osmosis, water following them. Chyme is often hyperosmotic, and so there is a pull of water into the lumen of the intestine from chyme, along with the movement of these electrolyte ions bringing water with them. In addition to water and bicarb and electrolytes, there are a large number of digestive enzymes that are secreted into the lumen of the small intestine. And we'll talk about those when we get to our little section on digestion. But there are peptidases that break up the small peptides that have been created from the other protein cleaving enzymes, for instance, pepsin in the stomach. into amino acids, which can be absorbed. There are enzymes that split disaccharides into monosaccharides, primarily glucose. And there is intestinal lipase that breaks fats into glycerol and fatty acids. Once the material, the chyme, has been fully digested and the nutrients absorbed, it reaches the ileocecal valve. The ileocecal valve is important for preventing the backflow of the bacterial-rich fecal contents into the small intestine. And there is a thickened layer of circular muscle that actually extends a little ways proximally into the small intestine, and it is tonically contracted, so it's generally somewhat constricted. There's not free emptying of material from the small intestine into the large intestine, and again, there is not backflow from the colon into the small intestine. And this is important for a number of reasons, one of which is to maintain chyme in the small intestine for long enough to be absorbed. When there's irritation of the colon, it inhibits peristalsis, it inhibits further emptying into the colon by maintaining a tonic constriction of the ileocecal valve. So let's talk a little bit more about digestion. Carbohydrate is ingested primarily as starches that have been created in nature by a chemical process called condensation, where a hydrogen ion is removed from a monosaccharide, a hydroxyl ion is removed from another monosaccharide, and they combine, forming disaccharides and chains of longer carbohydrate. During digestion, this is reversed. Chemical process is called hydrolysis, and the hydrogen ions and hydroxyl ions are added back to the polysaccharides, separating them into monosaccharides, so that we take disaccharides and also chains of carbohydrate that we call starch and return them to the state of being monosaccharides. Hydrolysis is also involved in the digestion of fats. So there are lipases, or fat digesting enzymes, that take a triglyceride molecule, return 3 molecules of water, splitting the fatty acids away from the glycerol. Proteins are bound by something called a peptide linkage, and hydrolysis is also involved in breaking peptide linkages, returning small chains, peptides, into their components, individual amino acids, releasing water in the process. So, sorry, that's how they're formed, releasing water. They are hydrolyzed, they are returned to amino acids by returning the hydrogen and hydroxyl ions to split them from each other. So there are 3 kinds of carbohydrate in the typical diet. The one that is probably ingested in the largest quantity is cellulose, and cellulose is not something that we have the enzymes to digest, and therefore it ends up passing through the gastrointestinal system and being eliminated with feces. It's fiber. The second carbohydrate is starch, which are these polymers of primarily glucose, and then disaccharides, which are things like table sugar. Carbohydrate digestion starts in the mouth and stomach. You remember I mentioned that salivary amylase, or ptyalin, is added to the masticated food in the mouth. It's from the parotid salivary glands, and it hydrolyzes some starches into maltose and other small polymers of glucose. But only a little bit of carbohydrate digestion takes place in the mouth. However, that enzyme remains active in the stomach, so as the stomach is mixing and creating chyme, about 30 to 40% of the starch digestion occurs here in the stomach, primarily to maltose, to disaccharide. Once the chyme reaches the small intestine, pancreatic enzymes are added. So there's a good deal of pancreatic amylase, which is a much more potent amylase than salivary amylase, and therefore, once the chyme enters the small intestine, within a very short time, the remainder of the carbohydrate is digested, and it's converted to either maltose or again other small glucose polymers, and passes to the duodenum. It's in the duodenum and the upper jejunum, at which point enzymes that are on the brush border in the intestinal epithelium take over. So there are 4 enzymes in the brush border in enterocyte villi that can split disaccharides such as lactase and sucrase and maltase and alpha dextrinase into monosaccharides, again primarily glucose. And the monosaccharides, they're all water soluble, but the at this brush border, as soon as the disaccharides are broken into monosaccharides, they're immediately absorbed into the portal blood. Proteins in the diet are long chains of amino acids that are bound by what are called peptide linkages. In the stomach, digestion of proteins begins with the secretion of a substance called pepsinogen, which is converted to pepsin, which is an enzyme that digests amino acids, preferably in a very acidic environment. So pepsin works best in the stomach where the pH is 2 to 3. Pepsin is the only proteinase that can digest the protein called collagen. The other peptidases that are going to be added in the small intestine can't do that. But pepsin only accounts for 10 to 20% of the total digestion of proteins. It takes long chains and breaks them down into smaller chains that are called proteoses and peptones and polypeptides. The vast majority of the digestion of protein occurs once the chyme has reached the small intestine and pancreatic proteolytic enzymes are added. So there are a number of enzymes that are secreted by the pancreas through the sphincter of Oddi that include trypsin, chymotrypsin, carboxypolypeptidase, and proelastase. The trypsin and chymotrypsin split the proteins into smaller polypeptides. The carboxypolypeptidase cleaves amino acids from the ends, and the proelastase is converted into elastase, which digests elastin. So we are digesting collagen, we are digesting elastin, which are in animal products and meat, and we are digesting other proteins down to smaller chains, polypeptides, and amino acids. As is true with the digestion of glucose, the final stage of protein digestion occurs on the microvilli, on the brush border of the enterocytes that line the villi in the small intestine. And those microvilli have peptidases. So the microvilli are protruding from the cell membranes and come in contact with the chyme and intestinal fluids, and they split the remaining larger polypeptides into either smaller polypeptides, dipeptides, tripeptides, and amino acids. The dipeptides and tripeptides are split within the enterocytes, and then the amino acids are passed out of the enterocytes into the portal circulation on the basilar side of the villi enterocytes. And then fat. The fat that we eat in our diet consists primarily of triglycerides, though there are also small quantities of phospholipids and cholesterol esters. In the stomach, a little bit of digestion of fat occurs, but just a little bit, because there is lingual lipase. So a little bit of lipase is in the saliva and works in the stomach. But the primary digestion of fats occurs in the intestine. And this is a little different from what we talked about with carbohydrates and proteins, which have very similar processes. In the small intestine, bile acids and lecithin are added to the chyme from the liver and the gallbladder through the bile duct and through the pancreatic duct and out of the sphincter of Oddi. And they function as surfactants that, along with the mixing that's occurring in the small intestine, physically breaks down the larger globules of fat into smaller globules, increasing surface area and allowing the digestive enzymes that are also being added to the chyme to act on the surface of those little globules. The vast majority of this mixing emulsification from the surfactants, bile acids, and lecithin occurs in the duodenum. So bile is really important to fat digestion. It allows the fat globules to be broken up by agitation and mixing, and decreases the diameter of the globules and therefore increases the surface area of these fat globules, which is important because the enzymes are water soluble. So they can only attack the surfaces of the fat globules. The fat globules need to be broken down considerably so that the enzymes in the aqueous phase of the chyme can attack those surfaces. Pancreatic lipase digests any triglyceride that it comes in contact to very quickly, so within a minute, leaving free fatty acids. The free fatty acids then combine with bile salts to make something called micelles. So micelles form from bile acids and monoglycerides and free fatty acids, which then transports them to the brush border of the intestinal epithelial cells. At that brush border, the free fatty acids are absorbed into the cells and then transported through the cell into the lacteals, which are those lymphatic vessels on the inside of the villi that transport them into the lymph vessels and eventually through the thoracic duct into the systemic circulation. But the bile salts are released back into the chyme, and some of those bile salts are reabsorbed and returned to the liver through something called enterohepatic circulation. So here we have a diagram of a micelle showing bile salts and then glycerol and fatty acids. Here the fatty acids are being broken off and released and absorbed into the brush border. The bile salts are left in the chyme, but eventually reabsorbed and returned to the liver through the enterohepatic portal circulation. Cholesterol esters and phospholipids are digested a little bit differently. A cholesterol acid, these are hydrolyzed by lipases that free the fatty acids, and then bile salt micelles are involved and help to transport them. Cholesterol is not absorbed without the function of micelles. So here we have again the large fat droplets with bile salts added, emulsifying, being digested into free fatty acids, forming micelles that are transported to the brush border, where triglycerides and cholesterol are absorbed, and ultimately the fatty acids are added to the lymph and lacteals. The cholesterol is absorbed into the capillaries. We have moved out of digestion and into absorption. All of the products of digestion are absorbed, but other things are absorbed as well. So we have to absorb water, and much water is absorbed by osmosis, and water can go in either direction depending upon the osmolality of the chyme. So if chyme is dilute, then water is absorbed into the intestinal membrane, into blood. When chyme is concentrated, water moves out through the intestinal mucosa into the chyme. Sodium is actively transported, and there are a couple of active transports. There is the sodium potassium ATPase that we've talked about a whole lot, but there are other mechanisms, and chloride is passively going along with sodium. The sodium concentration in chyme is similar to the sodium concentration in blood. If you remember, about 140 is normal. But the sodium concentration within the enterocytes is very low, just as it is in all other cells, because of the constant transportation of sodium out of the cell. And so sodium will move out of chyme into the cell down a concentration gradient, and then again is actively pumped out into the extracellular fluid. There are also carrier proteins that co-transport sodium along with amino acids and glucose, and we'll talk about those in a few minutes. And these are active at the brush border. Water follows sodium, and it's important to recognize that aldosterone, and we've talked about the action of aldosterone in the kidney, but aldosterone is also acting in the intestine to improve sodium and water absorption, to conserve sodium and water when a person's dehydrated. We lose almost no salt in the feces, but as we absorb sodium, we also absorb water and chloride and various other substances. Chloride absorption is quite fast. It diffuses. It's following sodium. It is also absorbed at the brush border in the distal small intestine, the ileum, and in the large intestine. And so it enters the cell on the luminal border and exits the cell on the basal membrane through channels. Bicarbonate is indirectly absorbed in the proximal small intestine, the duodenum and the jejunum. So when we're absorbing sodium, some of that is through an exchange with hydrogen ions. When sodium exchanges with hydrogen ions that are secreted into the lumen of the gut. The hydrogen ions combine with bicarbonate ions to form carbonic acid, which dissociates into water and carbon dioxide. So you've heard about this before. The water stays in the chyme, and the carbon dioxide, because it is both soluble in water and soluble in lipids, is rapidly absorbed into the blood and lost through the lungs. In addition to sodium and chloride, there is active absorption of a number of other ions. The absorption of calcium, primarily in the duodenum, is very tightly controlled, and it is tightly controlled through the mechanisms that we've talked about before, using parathyroid hormone and vitamin D. So you don't absorb any more calcium than you need. If you are getting an inadequate intake of calcium, it's going to ramp up production of parathyroid hormone, right? Low serum calcium makes you make parathyroid hormone, which is going to both get calcium out of bone but also promote absorption in the intestine, along with activating vitamin D, which is acting in the intestine to control calcium absorption. Iron is actively absorbed in the small intestine, and then potassium and magnesium and phosphate are all absorbed through the intestinal mucosa, not tightly controlled as calcium is. So I said at the beginning of absorption that we need to absorb all those nutrients. Once the carbohydrate is fully broken down into monosaccharides, it is co-transported. There are two mechanisms or two stages. First, you remember that the sodium potassium ATPase is depleting sodium inside the cells. It's pumping it out on the basal side. Therefore, sodium in the intestinal lumen is able to move through secondary active transport. It's secondary because it occurs because of that low sodium inside the cell created by the ATPase. And there are carbohydrate or glucose sodium co-transporters that move a sodium ion and a glucose molecule at the same time out of the lumen into the cell, where the sodium is pumped out, and at the basilar side it is passed into the bloodstream. Fructose is transported by something called facilitated diffusion that is not coupled with sodium transport. We already talked about how fats are absorbed, that they are dissolved. The monoglycerides and fatty acids are dissolved in micelles in combination with bile acids that are carried to the brush border and diffuse. The fatty acids leave the micelles and enter the epithelial cells. The micelles stay in the chyme, but the fatty acids and monoglycerides are processed in the enterocytes that create triglycerides that are released into the lymph channels, the lacteals that are inside the villi, and that join with lymphatic vessels that empty into the thoracic duct. There are small amounts of short and medium chain fatty acids that are absorbed directly into the portal blood. And then fat soluble vitamins, and it's important to know which vitamins are fat soluble. So vitamins A, D, E, and K being very important to blood clotting, are all incorporated into those micelles and all move with those other fats into the chylomicrons that are extruded from the cells into the lymph in the lacteals. And here we have a diagram showing Fatty acids and monoglycerides, again, being emulsified by bile acids, forming micelles, entering the brush border where the fatty acids are processed in the enterocyte into something called chylomicrons. So they are processed into new triglycerides, which are combined with proteins to make something called chylomicrons. And the chylomicrons are then extruded into the lacteals, which transport them away. I just put these pictures to demonstrate what's called lipemic plasma. So after a fatty meal, somebody who has blood drawn may have this milky plasma because of the chylomicrons that were sent through the lymph vessels directly into the systemic circulation by emptying at the thoracic duct into the vena cava. And this is an issue in the plasma industry. We don't want to infuse such fatty plasma into a patient. Water-soluble vitamins, including the B vitamins and vitamin C, are absorbed by co-transport mechanisms using the gradient of sodium. The exception is vitamin B12, also called cobalamin. Cobalamin binds to proteins in the saliva and in the stomach is transferred to the intrinsic factor that we talked about being secreted along with hydrochloric acid. This is a stable complex that then eventually enters the small intestine with the chyme and stays in the chyme to the terminal ileum, where there are cells that have a specific mechanism for absorbing vitamin B12. So if we don't make intrinsic factor, we don't absorb vitamin B12. If we don't have a terminal ileum or have a sick terminal ileum, we don't absorb vitamin B12. I, at one point, had the person in Mrs. Thomas's job was a woman who had had a gastric bypass procedure which eliminated her terminal ileum, and she ended up having very severe vitamin B12 deficiency that led to both anemia and to, in some cases, permanent nerve damage, because B12 is both important for blood formation and for health of the nervous system. So you absolutely need intrinsic factor to allow for absorption of vitamin B12. It combines with the dietary B12 in the stomach, which was first bound to a protein in the saliva. It is transported through the intestine to the terminal ileum, where there is a specific mechanism using pinocytosis to absorb it. And then it's transported through the portal circulation and stored in the liver. And again, B12 is extremely important both to make red blood cells, so hematopoiesis, and also for the health of nerves. We've gotten to the large intestine. The large intestine functions primarily to absorb water and electrolytes that are left in the material that's been extruded through the ileocecal valve out of the small intestine into the large intestine. And this allows that liquid material to be converted into solid feces in the proximal portion of the large intestine. And the large intestine, particularly the descending colon and rectum, also stores fecal matter until it can be expelled. So the absorption is happening primarily in the proximal intestine, where it goes from fluid to semi-fluid to mush to semi-mush. and then in the descending colon to a semi-solid, and ultimately to a solid material stored in the rectum and extruded. So remember that the main function of the large intestine is absorption of water. This requires mixing, as it does do the functions of the small intestine, which you remember the main function of the small intestine is digestion and absorption of nutrients. So in the large intestine, there are structures that are called haustra that are similar to the segmentation contractions in the small intestine. There are long strips of longitudinal muscle that are called tenei, coli, or the colic band, and a combination of contraction of the circular muscles and the longitudinal muscles causes a bulging that is called haustra or haustrations. As occurs with mixing contractions in the small intestine, the areas that are involved move from one part of the colon to another part of the colon. And here we have some x-rays. The x-ray on the left shows a lot of gas in the large intestine, but you can see haustra. And the x-ray on the right is a barium x-ray. So there's barium here, and I am wondering if this is a reversed barium enema. I think it is. But you can see where circular smooth muscle contractions are forming haustra. Now, in the large intestine, there are no villi. Remember, there were villi in the small intestine, but there are no villi in the large intestine. There are lots of crypts. There is not significant secretion of digestive enzymes, so we're no longer digesting our material. There are cells that secrete mucus, which does two things. One is that it lubricates, allowing for passage of feces without significant damage to the epithelium, but also the bacteria that are an important part of the function of the large intestine create acids, and so there is bicarbonate. This is an alkaline mucus, and it helps again to protect the wall of the large intestine from the bacteria and the acidity created by the bacteria. Most of the water is absorbed in the proximal half of the large intestine. The distal colon is the storage colon and stores feces until excretion. The mucosa, when it secretes bicarbonate ions, is absorbing chloride ions. There's an exchange of bicarbonate for chloride. But there is also absorption of sodium that creates an osmotic gradient that causes absorption of water across the intestinal mucosa. Again, you remember that sodium potassium ATPase is creating a very low sodium environment inside the cells of this epithelium. So the water in the lumen is going to go with sodium that is being absorbed from the lumen, and then the sodium is going to be excreted and the water is going to be absorbed into the circulation. So there's only so much water that can be absorbed in the large intestine, and if there's too much water to be absorbed, it appears in the feces. But there are also certain bacterial infections and certain toxins created by bacteria that can cause excess secretion of water into the lumen, which results in diarrhea. Under normal circumstances, feces are about 3/4 water and about 1/4 of solid matter comprising bacteria and fiber and fat and minerals, inorganic matter, and protein, a little bit of protein. So a little bit of fat, a little bit of protein that haven't been digested. There are propulsive movements in the large intestine, and some of those propulsive movements are called mass movements. So it takes time for the chyme to get through the small intestine. Here it says 8 to 15. I think earlier it said 3 to 7. Oh, never mind. 8 to 15 hours for the chyme to get from the ileocecal valve through the colon to the rectum. And after meals, there are propulsive movements that are called mass movements, usually 1 to 3 a day. And once a mass movement pushes feces into the rectum, triggering distension, then defecation occurs. There is also a reflex, an important reflex called the gastrocolic reflex. When the stomach is distended by a meal, then there are signals that are mediated by gastrin and cholecystokinin that are detected, these hormonal signals, in the colon, triggering motility in the colon, mass movements that stimulate rectal filling that leads to defecation. Defecation involves both smooth muscle and skeletal muscle, and this is something that's pretty important. As the rectum is filled by these propulsive movements, there is a smooth muscle internal sphincter that relaxes, but the external skeletal voluntary sphincter stays contracted. Once the rectum is full enough, we have the urge to defecate. We take ourselves to an appropriate spot and we relax the external sphincter, and smooth muscle in the rectum then contracts and pushes the feces out through the anal canal. And we typically add what's called a Valsalva maneuver, which is we increase intra-abdominal pressure to push the feces out through the anal canal. And we poop. Dr. Perry really likes poop. I wouldn't be surprised if there was a test question about pooping. Finally, I want to talk a little bit about some of the organs that we would call accessory organs in the GI tract. These are the organs that often add the things that I've talked about being added. So they include the liver and the pancreas and the gallbladder. And here we have a picture of the liver with the hepatic ducts that lead to the common bile duct and to the gallbladder, and the pancreas with the pancreatic duct. They join, and the head of the pancreas and the material is pushed into the lumen of the duodenum at the sphincter of Oddi. I think that's how you say it. I meant to ask Mr. Google that. So the liver is really important. The liver does tons of things, but when we're talking about digestion in the gastrointestinal tract, one of the important things that the liver does is that it processes bilirubin. Where does the bilirubin come from? It comes from the degradation of heme that is basically processed from destroyed red blood cells. Red blood cells have a finite lifespan, so the heme from dead red blood cells becomes bilirubin and is conjugated in the liver to form water-soluble conjugated bilirubin. This is also called direct bilirubin when you're in lab medicine. So conjugated bilirubin has been processed by the liver. The conjugated bilirubin is a big part of bile and is important in the digestion of fat, and we've talked about that. Now, in the gut, some of that conjugated bilirubin is deconjugated by assorted enzymes, and it's metabolized into a substance called urobilinogen that's reabsorbed and returned to the liver. But some of it doesn't get picked back up by the liver, and urobilinogen is the pigment that makes urine yellow. And then some of it in the gut, again, is deconjugated to urobilin and then something called stercobilin. And these are excreted with the feces, and they are the pigment that makes poop brown. So, bile, you might not have thought about this, but bile is what makes urine yellow and makes poop brown. You remember that the liver stores carbohydrate as glycogen. So when we are absorbing large amounts of carbohydrate through the portal circulation, it goes to the liver where excess is stored as glycogen. But in circumstances of fasting and low blood sugar, that glycogen is broken down into glucose. But in addition, the liver can take other molecules like amino acids and fatty acids and make glucose, and this is called gluconeogenesis. It synthesizes non-essential amino acids. There are some amino acids that we have to ingest, but there are some that the liver can kind of mess around with and make. The liver converts ammonia to urea, which is excreted in the urine. And when the liver isn't working, we can have excess circulating ammonia, which results in central nervous system dysfunction that's called hepatic encephalopathy. When there's too much ammonia, ammonia is lipid soluble. It easily crosses the blood-brain barrier. And the condition hepatic encephalopathy, which often occurs in people with end-stage cirrhosis, causes confusion and lethargy. It causes a certain kind of tremor called asterixis, and it can lead to coma and to death. The liver also oxidizes fatty acids. It synthesizes cholesterol and phospholipids and lipoproteins that are important in the production and maintenance of cell membranes. It synthesizes a ton of proteins. So the protein that is the most significant in quantity, I can't say the most important, but the albumin we've talked about accounts for plasma oncotic pressure. And you remember that edema can occur when we have insufficient albumin, and that can happen when either the liver isn't working and isn't making albumin, or when the kidneys aren't working and are losing too much albumin. But the protein albumin is synthesized in the liver. There are a number of proteins that are called acute phase reactants, like C-reactive protein, that are made in the liver and that help in our response to stress. There are a ton of hormone binding and hormone transporting proteins, and I believe we've already talked about some of those in endocrinology. Many hormones bind to proteins to be transported, at least partially. And there's thyroid binding globulin. Globulin is a word for many kinds of proteins. The liver also makes clotting factors, and clotting factors are proteins that are crucial to allow us to clot our blood, but also to break down blood clots to prevent us from turning into one big blood clot. And that's all dependent on the liver. Obviously, blood clotting also uses the cellular elements that we call platelets. People know that the liver is an organ that detoxifies, and it does that by metabolizing substances. So many drugs are metabolized, which results in something called a first pass metabolism. Drugs that are ingested have to go through the liver, and that can affect whether they are activated or inactivated, how much of their activity continues after the first pass of the liver. Remembering that everything that's absorbed from the GI tract goes into the portal circulation, meaning it passes through the liver. There are enzymes in hepatocytes that modify drugs, but also toxins, both toxins that are created by metabolism and also exogenous toxins that change those so that they can be eliminated either in the urine or in bile. So there are a number of enzymes that we call the cytochrome P450 enzymes. And I'm sure that you have already talked about these in pharmacology, because this is one of the early topics in pharmacology, is what happens to drugs after you ingest them. So they can be oxidized or hydroxylated, and there are a number of other potential reactions that are catalyzed by the cytochrome P450 enzymes. Those are called phase 1 reactions. Then there are phase 2 reactions, and again, this is toxins and drugs. The molecules can be conjugated with glucuronide, I can't say that word, glucuronide, sulfate, with amino acids, with glutathione. These are phase 2 reactions. And then finally, many lipophilic substances that enter the portal circulation are made water soluble, often by the things that we talked about above, these reactions: oxidation, conjugation, and then they are eliminated in the bile through the gut. I'm going to review what bile does because it's just so important. It's a surfactant, it's a detergent, it's a soap. So the bile acids in bile help to emulsify big globs of fat into tiny globs of fat, increasing the surface area so that lipase from pancreatic secretion can attack the outer surface and digest those lipids into their components, fatty acids and glycerol. The bile acids also assist with absorption through the intestinal mucous membrane. And in addition, we understand that it is a surfactant that aids in fat digestion, but it's also a really important way that waste products from blood breakdown are eliminated. So bilirubin and excess cholesterol. The bilirubin is a product of the breakdown of heme from senescent red blood cells, and then excess cholesterol is used to synthesize bile acids. There are 2 stages of bile secretion. So it is made by hepatocytes, and in the first stage it is secreted into these tiny bile caniculi between hepatocytes. In that initial bile, there's a lot of bile acids, cholesterol, some other organic substances. The canaliculi flow towards septa between the lobes, so they empty into bile ducts and then into bigger and bigger bile ducts, finally reaching the hepatic duct, which is the main bile duct in the liver, and the common bile duct. And then bile can be diverted and stored in the gallbladder, so it's diverted through the cystic duct, or it empties directly along with pancreatic secretions into the duodenum. The storage in the gallbladder can be minutes to hours. As the substance that was initially made in hepatocytes is traveling through the bile ducts, there are further secretions that are added to this liquid substance we call bile. So there's a solution of water with sodium and bicarb ions that doubles about the quantity of bile. And that watery electrolyte solution is stimulated by the hormone secretin. The gallbladder can store between 1 and 2 ounces of bile. So storage is limited as bile is made. And I believe we said that the liver secretes about a liter of bile a day. The bile itself is made up of bile salts, which are the primary surfactant. There are also bilirubin, cholesterol, lecithin, obviously water and electrolytes. As as the gallbladder stores bile, some of the water and electrolytes are reabsorbed. So the bile that is stored in the gallbladder is also concentrated in the gallbladder. And this is a Medscape slide showing you what's in chemically what's in bile, not discussing the water. When we eat and food enters the upper GI tract, so the stomach, and is eventually gets to the pylorus and is ejected into the first part of the duodenum, the gallbladder empties. And if you remember, cholecystokinin, which is a hormone that is made in a couple places, one of the main places is cells in the upper duodenum, causes the gallbladder wall, which contains smooth muscle, to contract. So the contents of the gallbladder are pushed into the common bile duct and then join with pancreatic secretions. And the sphincter of Oddi has to be relaxed at the same time. So CCK, cholecystokinin, responds to a fatty meal by contracting the gallbladder and releasing the sphincter of Oddi. And it takes emptying doesn't occur nearly as well when there's not fat in the food. So a meal that is a fat-free meal will not cause the release of cholecystokinin and contraction of the gallbladder. On the other hand, I'm sure you've all heard of people with gallstones whose pain starts, and sometimes you have really severe pain after eating a fatty meal. So they eat a hamburger and fries from McDonald's, and before you know it, are doubled over and vomiting with the pain because their gallbladder tried to contract and stones were obstructing the outflow of bile, and this is a very painful thing. And so people with gallstones often find themselves trying to avoid fatty meals. Bile is made from cholesterol. Bile salts are made from cholesterol. So cholic acid and chenodeoxycholic acid salts are what we call bile salts. In the intestinal tract, and we're talking here in the duodenum, in the first part of the small intestine, they act as detergents to emulsify the fat and decrease the surface tension on the big globules. And there is mixing, as we've talked about in the small intestine, and the big fat globs are broken up into small fat globs. So bile is required to help absorb fatty acids, monoglycerides, and cholesterol and phospholipids and other lipids. We've already talked about micelles and how micelles are the way that bile salts and digested fats, so fatty acids, the lipids that are the product of digestion, along with bile salts, are carried to the villi and microvilli on the intestinal mucosa, and they are absorbed, processed in the enterocytes, and then absorbed not into blood but into the lymph, which drains ultimately into the blood. Now, the bile salts that were part of those micelles, much of those bile salts get reabsorbed in the ileum, in the last part of the small intestine, and then return to the liver via the enterohepatic circulation. We already talked about, when we were talking about fat digestion, I believe, how some of those bile salts get further processed by intestinal bacteria into urobilin and stercobilin, which give color to poop and makes stool brown, but also some of them are absorbed into the circulation and end up in the general circulation and give the yellow color to urine from urobilinogen. And you'll see urobilinogen as on a urine dipstick. It's one of the things that we are looking at, as well as bile itself. Pancreatic secretions are obviously super important in digestion, and again, we've talked about them briefly, but let's go back to the anatomy of the pancreas. You remember that the pancreas has endocrine cells, but it also has exocrine cells, and those exocrine cells are grouped into something called acini. They secrete lots of a sodium bicarbonate rich aqueous solution, which dissolves the enzymes that are also secreted, and they all together flow from the acini in the pancreas through the pancreatic duct, joining with the common bile duct and exiting through it says the duodenal major papilla, which is also what we've been calling the sphincter of Oddi. Pancreatic enzymes and solution are secreted in response to the presence of food in the first part of the small intestine and the types of food in the chyme that's coming through the pylorus into the duodenal bulb. So lots of messaging going on here. Some of it hormonal, being picked up in the general circulation, some of it local, so paracrines. So in pancreatic juice, if you want to call it that, there are a number of digestive enzymes. There are enzymes to digest protein. The primary one is trypsin, but there is also chymotrypsin and carboxypolypeptidase. There are enzymes to digest carbohydrate, and that's pancreatic amylase. So you remember that a small part of digestion, or a minority of the digestion of carbohydrates, occurs from the salivary amylase being processed at low pH in the stomach as the food is mixed and turned into chyme. But by the time it hits the small intestine, pancreatic amylase is very efficient at further breaking down those carbohydrates into much smaller molecules. There are enzymes to digest fat, primarily pancreatic lipase, and it acts on the fat that has been emulsified by the bile acids into fatty acids and monoglycerides, and creates micelles. Cholesterol esterase hydrolyzes cholesterol esters, and phospholipase breaks down phospholipids. The bicarbonate solution that's made by the pancreas is really important to neutralize stomach acid. So the enzymes of the pancreatic secretion are secreted by glands, but they're exocrine glands. The water and the bicarbonate are secreted by the epithelial cells lining all of the pancreatic ducts. When there are hormonal or neural impulses stimulating the release of pancreatic juice, the bicarbonate concentration increases dramatically, up to 5 times the levels of bicarbonate in plasma. And that alkaline solution, again, is crucial for neutralizing the hydrochloric acid that enters the duodenum from the stomach through the pylorus. So the things that trigger pancreatic secretion include acetylcholine from the autonomic nervous system and also from the enteric nervous system, along with the hormones cholecystokinin and secretin. And here are your references, and I hope that this has been helpful and that you will spend some time kind of reviewing this, but also give me feedback about its organization, because I did modify that a little bit this year in response to feedback last year.